So my name is Nat Michaels. I'm a rising senior at Reed College, and this summer my project was identifying and annotating long non-coding RNAs in eggplant and tomato. So basically, a long non-coding RNA is an RNA that has little to no potential to be translated into a protein, is longer than 200 nucleotides, is expressed at a tissue level or very low levels of expression, and can play important functional roles and is also classified based on its position in the genome. So this summer specifically, we looked at intergenic, which is where they're between coding genes, and antisense transcripts, where they're transcribed in the opposite direction as a known coding gene. And we did this basically because they're the easiest to differentiate from a coding gene. And so you might be asking, why tomato, why eggplant? What's the importance of this? These are both really important food crops. They're top 30, both for metric tonnage produced in the world. And these are also the cultivars that we chose to sequence are also two of the most studied cultivars for tomato and eggplant in science. So this would provide not only an important scientific resource for future research, but also my lab, the Frank lab, is interested in grafting and how we can use grafting to improve things like food crops. Like, how can we send RNAs across that are going to make fruit bigger or something? So eggplant and tomato are graft compatible. So we also know that non-coding RNAs have high potential to be mobile. So if we identify these long non-coding RNAs in tomato and eggplant, the hope is that we'll be able to identify them later on if they are mobile. And so in the end, we ended up identifying about 6,000 high confidence transcripts for eggplant and 3,224 for tomato. And yeah, I mean, the rest of these figures are basically just saying that our data looks like what we expected. We saw shorter lengths than mRNAs. We saw lower levels of conservation. We saw tissue specificity in eggplant. We didn't have the data to do tissue stuff in tomato, but basically, yeah, our data all looks good. So hopefully this will provide an important resource for later science. And thank you to everyone for involving me in this project.
Great. Thank you so much. It's always very important to provide resources for the greater community. So very nice.